A study titled Gene Therapy for Red-Grain Color Blindness in Adult Primates was published in 2009 and was collectively conducted by a group of scientists. This study provides evidence to support the concept that gene therapy can be successful in restoring color vision to monkeys. Not only are humans commonly affected by color blindness, but this disorder also affects primate species. For the study, an experiment was conducted. The scientists used squirrel monkeys as their test subject. The aim of the study was to endeavour to restore trichromatic vision in adult primates using gene therapy. The methods that were used during the study comprised of the scientists acquiring adult squirrel monkeys that lacked the L-opsin gene. Scientists sought to solve the issue of these primates missing the L-opsin gene. This was achieved over a time period of 20 weeks by giving the squirrel monkeys 3 different injections into the retina part of the eye. The experiment was successful as the scientists found that after 20 weeks, the squirrel monkeys had developed the presence of the new pigment in the photoreceptor. This was apparent as the scientists tested the colour vision in the monkeys as shown in the following picture. Behavioural tests were conducted, and this involved recording the monkey's reaction towards different coloured pictures. Depending upon the monkey's vision, the monkey was either dichromatic, colourblind, or trichromatic, normal colour vision. Over time, the monkeys eventually developed the ability to differentiate between the different colours. The results of this colour test are represented in the following graph. Figure A clearly shows the squirrel monkey's threshold comparison over time. The arrow represents the 20 weeks after treatments. This arrow shows that there was a significant drop in threshold after the 20 weeks. This is shown by the solid line joined by the blue dots. The trichromatic female threshold is represented by the solid line that has small green dots. From this graph, it can be concluded that the monkeys gradually developed better vision as they were able to differentiate the 4 various colours from the grey background. This was recognised by the scientists from the use of the colour tests. These 2 graphs represent the records of 2 different monkeys. Figure B displays information obtained from monkey 1. Figure C demonstrates the findings related to monkey 2. These graphs visualise the comparison between pre- and post-injection threshold results of the dominant wavelength, which is measured in nanometers. There is a related article titled Chromatic Coding from Cone-type Unselective Circuits in the Mouse Retina. This study was conducted by L. Chang, T. Broniger, and T. Euler. The difference between these studies is that the scientists used mice as their test subject and not adult primates. In conclusion, this experiment was successful in demonstrating that it is possible to restore colour vision in adult primates. All that is required is the existence of a third cone type. Based on the results of this study, it can be concluded that the adult monkeys now have a trichromatic colour vision as a result of gene therapy. This study concludes that with the use of gene therapy, there is a possibility of curing colour blindness in humans.